Hello, my name is Jean-Michel. I am a professor at the University of Wisconsin-Madison. Thank you for giving me the opportunity to talk about our projects to try to improve the benefits of biological nitrogen fixation for cereal crops. Most plants get their nitrogen from the soil, mostly in the form of nitrate and ammonium. Nitrogen fixation is the process of converting dinitrogen from the air into ammonium. And industrial fixation, also called the Haber-Bosch process, is the process by which we produce synthetic fertilizers using natural gas as a source of energy. And it's estimated that about half of the world population is currently sustained through the use of these synthetic fertilizers. At the same time, I'm sure you have heard about the negative consequences of these fertilizers, such as the conversion of these fertilizers into greenhouse gases like nitrous oxide, but also the nitrogen leaching and nitrogen losses in the soil. These synthetic fertilizers are absolutely essential for cereal crops in particular that require a large amount of nitrogen inputs. Today I'm going to talk about biological nitrogen fixation performed by bacteria and archaea, and legumes in particular are able to associate efficiently with some of these nitrogen fixing bacteria called rhizobia. That's why you have legumes in many of our cropping systems around the world. They require very little, if any, fertilizer inputs, and they can even add nitrogen to the system through processes like crop rotations. Unfortunately, most of our cereal crops, at least the ones we are using now, associate very poorly with nitrogen-fixing bacteria. And so it had been a goal of the scientific community for more than 100 years to try to improve association between nitrogen-fixing bacteria, sometimes called diazotrophs, and cereal crops. Some approaches are more on the microbial side, and some approaches are more on the plant side. Historically, the first approach that has been followed is to use the root nodule symbiosis system of legumes and to transfer it to cereals. So that many researchers have been working on that kind of project, and I think it's still a very valid approach, in particular given the progress over the last decades and trying to understand the plant genes that control root nodule symbiosis and the evolution of this trait. And many labs, including my lab, are still working towards this goal of engineering root nodule symbiosis in non-legume crops. Another approach, but in that case here entirely on the plant side, is to get rid of the microbe and express the nitrogenase enzyme in plant cells, whether in plant mitochondria or plant chloroplasts. Again, many colleagues are working towards this goal. It's a great goal, very important research, but both of these approaches, unfortunately, are really long-term. So today I wanted to focus on and talk about two approaches that are more short-term. One of them is entirely on the microbial side, the idea of isolating diazotrophs associated naturally with cereal crops, maize or sorghum, and to genetically modify these bacteria to fix nitrogen at high rates, even when nitrogen is present in the environment, which is often the case in agricultural systems, and also to release that nitrogen outside of the cell. So I apologize for the very busy slide, but I wanted quickly to show you what are the main targets of our genetic engineering. The first target are the regulator of NIF gene expression, in particular the NIF-L, a negative regulator of NIF gene expression, and NIF-A, which is a positive regulator. So we try to knock out NIF-L and overexpress NIF-A, and that leads to higher NIF gene expression and nitrogenase activity. A second target that we work on is the glutamine synthetase. We reduce the ability of these bacteria to assimilate ammonium, and so ammonium is going to build up inside of these cells. A third target are the ammonium importers. We prevent these bacteria from recapturing the ammonium that leaks out of the cell. So by doing all that, we and other labs engineered over the years ammonium excreter bacteria. The main issue with this approach is fitness, that not surprisingly, many of these ammonium excreters have a fitness defect. And here I'm showing you a growth curve of a wild-type Klebsiella varicola, and we here with different colors. These are different mutants we made, ammonium excreting mutants. And that fitness decrease of all these mutants leads to a reduced ability to colonize the plant and also poor competition with other microbes, in particular in the rhizosphere. And so one approach is to use synthetic biology to try to engineer inducible fixers that, for instance, would only fix in the presence of the plant. And my lab and other labs also, I know, are also working towards this goal. A second approach is try to understand better microbe in the rhizosphere. Interaction with fungi, like mycorrhizal fungi, are usually beneficial because these fungi help transferring ammonium from the ammonium excretors to the plant. Interactions with bacteria are more complex. Sometimes it's competition, and sometimes it's more synergy, where interestingly, we see that the effect that other bacteria have on the nitrogen-fixing bacteria on nitrogen fixation or growth are often independent. For instance, we can have some bacteria that promote growth without affecting nitrogen fixation, or promote nitrogen fixation without affecting growth, which is the kind of bacteria that we're very interested in. And we try to understand the mechanisms of these synergies between microbes. Second approach I want to talk about is the approach of having a better plant. In many ways, this approach is similar to engineering root nodules. We want a better host for nitrogen-fixing bacteria. But the idea here is to explore natural diversity. Instead of re-engineering a whole new organ in an intracellular symbiosis, we explore the possibility that some accessions of our crops, like maize or sorghum, some accessions may be better at hosting these bacteria than our current accessions. And that line of work led me to characterize accessions of maize and sorghum in particular that have roots on the stem. These roots never touch the ground. We call them aerial roots to indicate that they stay in the air. And when it rains, these roots produce a gel, a mucilage, that is a really good place for nitrogen-fixing bacteria to fix nitrogen and to deliver the nitrogen to the plant. And so that project has been going on in my lab, both initially in maize and then rapidly in sorghum, and hence my weird maize-sorghum hybrid I'm showing you here. And to summarize, what we have found is that we published in 2018 that maize accession with that kind of trait can get more than half of their nitrogen from the atmosphere. We published in 2025 that sorghum accessions with this trait obtain about 40% of nitrogen from the atmosphere. We identified genes using QTL and GWAS approaches. And of course, we have started the breeding effort to introgress that trait into accessions that we can use in temperate regions, the US, but also many places around the world. And of course, we are interested in molecular mechanisms, trying to understand what are the genes that control the production of that mucilage in particular. I would like to finish to conclude and to come back on these 4 different approaches and answer 2 questions that people often ask me. One of them is, are we there yet? When are we going to see products on the market delivering that kind of solutions? So for the bacterial side, we already have products on the market commercialized, in particular by companies like Pivot Bio and soon others, I guess, where Pivot Bio, for instance, has remodeled bacteria that they deliver nitrogen to maize, sorghum, cotton, wheat. So we are there already. There are some of these products, and I expect that the field is going to make more progress with better manipulations and possibly some of these helper bacteria. For the plant side, the plant trait, and in particular that aerial root trait, I would say we hope that within about 4 or 5 years maximum, we'll have varieties that will be available to growers. And that is the goal of, in particular, an ARPA-E grant that my lab and others obtained recently to really try to deliver products for growers in the short term. All right. And the second question is, how much nitrogen are we talking about? So the bacterial technology alone, at this point, probably delivers about 15 to 20% of nitrogen from the air. And here I'm citing a paper from the Pivot Bio company, and I'm a co-author on that paper, where we estimate that the product, the current product of Pivot Bio delivers about probably 15 to 20% of nitrogen from the atmosphere. The plant trait is actually even more powerful. We think that the accessions of that where we introgress the trait obtain about 35 to 50% of their nitrogen from the air, and we have data on that in maize and sorghum already. All right, I'd like to conclude and thank people from my lab. Of course, in particular, lab members Valentina Infante, Claire Palmer, Rafael Venado, Rubens Diogo, Tess Kern, and Junko Maeda. My collaborators at UW-Madison in Florida, Georgia, and at Duke University, and funding from DOE, including the GRBRC, USDA, NSF, Pivot Bio, and the Wisconsin Corn Grower Association. Thank you very much for your attention.
Hello, SoilCon. My name is Mallory Choudoir, and I am an assistant professor and soil microbiome extension specialist in the Department of Plant and Microbiology at North Carolina State University. My applied research program brings a microbial community ecology perspective, and we leverage different molecular sequencing approaches and microbiome science to generate insights that support sustainable agriculture and climate change resilience. So we do this by evaluating commercial microbial products, developing microbiome-centered solutions, and extending knowledge to diverse communities of interest. And we have the opportunity to work across diverse cropping systems from small to large scale and from row to specialty crops. So this work, this idea is foundational to our work, that the principles to manage soil health also support healthy microbiomes, minimizing disturbance, maintaining living roots, maximizing soil cover, and increasing ecosystem level biodiversity are all practices that support and build healthy soil microbiomes. So this is really important, building and supporting resilient soil microbiomes, especially in the face of climate change and increasing extreme weather events. All of these stressors threaten to further disrupt soil microbiomes and their important critical agroecosystem functions. Today, I'm going to briefly share two projects we are working on that address soil health challenges in North Carolina, aiming to build resilience into these ecosystems. These challenges include building soil organic matter in these low carbon sandy soils in the coastal plain, and then also managing ecosystems that are experiencing soil salinization due to saltwater intrusion on the East Coast. So for this first study, this is a rapid soil health study with an objective to evaluate carbon input strategies during a 3-year organic transition project from conventional to organic management in a high disturbance crop rotation. So 2 field trials were installed at NC State research stations in the Sandy Coastal Plain region, Upper Coastal Plain Research Station, and Horticultural Crops Research Station. This study was a 3-year corn, soybean, and sweet potato rotation with cover crop and no cover crop plots, and also either a single amendment addition at the start of the study or amendments applied annually. And the amendment strategies included a control no amendment, biochar, compost, and a mix of 50-50 biochar and compost. These images on the left are aerial views of these plots after the installation of this trial. And this work was led by the Woodley Lab and graduate student Fred Teasley. So now I'm going to share some general trends looking at soil organic carbon across the study years sampled in the top 15 centimeters. So here in these plots, we have on the top row the no cover crop plots, and on the bottom row, the plots with cover crops. In the control, so no amendment plots, we see at year 3 there is a significant signal in SOC between the cover crop and the no cover crop plots. So we see an increase in carbon at year 3 in the cover crop plots. When we look at the different application rate amendments in combination with cover crops, we see this signal between cover crop and no cover crop earlier in year 2. And then finally, by year 3, there was no difference in total carbon stocks between the application timing of the single or annual. And what my team is doing is characterizing the microbial biodiversity, microbial community function, and microbial community resilience. So the legacy impacts of these different amendment strategies. So some preliminary data from our work, we have found that bacterial diversity differs between sites as well as amendment. So in this plot, we have increasing biodiversity on the y-axis, and we see this difference in biodiversity between these two locations. If we look within location, we see additional patterns where the amendment is also explaining differences in bacterial biodiversity. So in this plot, we have the shapes designating the different locations and then the colors designating microbial diversity at these different amendment and application rates. Finally, we also see that bacterial community composition is largely driven by location, but within location, we see variation explained by amendment. So in this plot, each point is a bacterial soil community, so the types of species that are present and their abundances. And points that are closer together are more similar in composition, and points that are further away are more dissimilar. So here we have the location designated by shapes, and we see this distinct clustering based on circles or triangles. And then if we look within location, we see additional clustering driven by amendment and application timing. So the take-home messages from this study so far are that conversion to organic from conventional does not guarantee increased soil health. Cover crops plus organic amendments can build soil carbon more rapidly than either of these practices alone. And from a microbial perspective, even in locations with very similar climate and soil type, microbial diversity will differ. And amendment patterns, both application and amendment strategy, shapes patterns of microbial diversity and function within a given location. So we are excited to share lots more findings from this study. But for now, I'm going to move on to our second project looking at sea level rise and saltwater intrusion, which is causing soil salinization and threatening these vulnerable soil ecosystems on the East Coast. This is in collaboration with Dr. Matt Ricker in Crop and Soil Sciences at NC State and Andrea Gibbs, NC State Extension Agent. So we are working to characterize soil health metrics and microbial communities across these dynamic salinization gradients. And this is important because this salinization is resulting in significant crop as well as economic loss in these coastal communities. So we have been sampling soils and characterizing soil health and community microbiome communities across different salinity gradients, including this gradient across a game land conservation preserve. So we have sampled the marshland right on the coast, the ghost forest, which is an ecosystem that has experienced soil salinization, and the above ground plants are no longer able to survive. So there's a lot of above ground dead carbon. And then a swamp forest, which has not yet been turned into a ghost forest. And we find that microbial community composition varies across these gradients. So here we have a plot looking at bacterial biodiversity. Again, in these plots, each point is a single soil community, and points that are closer together are more similar. So we see unique communities of soil microbes in this marshland, the ghost forest, and the swamp forest. And these differences are explained by differences in soil carbon and other soil characteristics. We also see a pattern, not as strong, but a similar pattern in the fungal communities. So we are exploring the relationship between the different soil health metrics and microbial community composition and function across these changing ecosystems. And our goal is to harness these salt adapted communities and see if these are potential targets for biostimulants to improve salinity tolerance for growing crops under these increasing soil salinity conditions. So with that, I'd like to thank the members of my lab, my NC State collaborators, and our funding sources. Thank you.
Greetings, I'm Kirsten Hofmockel. I'm a chief scientist at the Pacific Northwest National Lab. And today I want to share with you our research on leveraging microbial function to manage soil organic matter and nutrient cycling in soils. So the work that we do is focused on soil stewardship for societal well-being. And so we're thinking about all the important roles that soils play for our national security, for climate resilience, and for economic prosperity. And the big question that we want to understand is how microbes can promote soil health, leading to secure, profitable, and resilient ecosystems. And so there's been a major understanding and advancement in soil microbiome and its benefits to soil health. And so we know that microbes generate things like EPS that increase, that's extra, these are biofilms that microbes make on mineral surfaces. This can help with soil water retention. We know that microbes are a major plant carbon sink in that they can absorb and assimilate exudates that come from the plant and then turn them into biomass that persists within the soil. They contribute to carbon sequestration through their death and necromass that persists in the soil. And then finally, they can contribute to plant growth promotion, both by generating nutrients and also through signaling molecules. And so one of the major research frontiers for managing soil microbiomes is to control soil organic matter and nutrient cycling. And we want to understand how these microbial interactions in the microbiome with the rhizosphere and the soil can regulate nutrient cycling and soil organic matter formation. And so this is a conceptual model on how we think about plants feeding the microbes through their roots. This increases microbial metabolism that we can measure microbial activity through the respiration. That's sort of how they breathe, how plants and microbes breathe. We know that microbes can grow and they like to grow on surfaces, and that's where they might make these biofilms that help with water holding capacity and help with aggregation and soil structure. As those microbes die, some of that material gets recycled and other microbes can live off of it. And a large portion of it actually persists in the soil. So as the microbes die, if they're associated with soil surfaces or mineral associated organic matter, it can make this complex and then persist in the soil. And so in the field, we're out there and we can look at that respiration that I said is that the breathing life of the roots and the microbes in the soil. And so here are our undergrads measuring that respiration. And we did this over an entire year to look at how respiration changes in the field. And we see that it largely reflects root biomass and root activity. And so we really want to understand how are those roots interacting with the microbes to increase nutrient availability in soil organic matter. And so one way to do this is to look at metabolipidomics. And so this is a high resolution molecular technique that lets us understand what the roots are making and what's persisting in the soil. And so here we're looking at bare soil and comparing it to bulk soil, root associated soil, and a root wash. And so what we find here is that when we are close to the root, the metabolites primarily function with the active microbes. They can be signals to the microbes to foster beneficial microbes. There's also things like fatty acids or secondary metabolites that are interacting with the microbes. We also see these organic acids that are abundant in root exudates, and they play a key role in nutrient cycling by solubilizing some of these mineral-associated nutrients. As we get farther away from the root, or we look in the bare soil, we see a high concentration of molecules that are associated with carbon storage and long-term soil organic matter formation. And so, for example, we see these phospholipid fatty acids, microbial phospholipids. So we know microbial cells are there. It demonstrates we've got growth in this bulk soil, but it's also associated with death of these microbes. We see there's these triacylglycerols. So these are storage molecules that can serve as carbon stores. And these TGs can persist in the soil and be broken down by microbes when energy, more bioavailable forms of energy are less abundant. And finally, we see these very long chain fatty acids. And these are precursors to protective structures like suberin that you would find in roots or waxes that you might find in leaves. And in the bulk soil, they're very slow to decompose. And so they contribute to more slow cycling soil organic matter. And they also increase stability and soil water retention properties. So when we looked then deeper at how microbial respiration might reflect these substrates, here we're using substrate-induced respiration, and we took soil from the field over each of these months, fed the microbes all sorts of different substrates, a panel of substrates that were representative of those metabolites. And we see that there's a variability over the seasons, but what's really interesting to us is that even in the winter months, we see high microbial activity. And so these were perennial tall wheatgrass plots. And we see that in the winter months, there's high decomposition of chitin. This is basically like fungal biomass is getting turned over. It's rich in carbon and nitrogen. So it makes nitrogen available in the spring when the weather warms up, and then that cycling slows down. We find small bioavailable molecules like glucose and other small sugars to be abundant throughout the growing season, and microbes are always able to take up and use a high quantity of those bioavailable molecules. We find these fatty acids that I talked about previously. They can originate from decomposition of these plant materials or breakdown of those triacylglycerides that I talked about. The microbes are breaking those down during winter months. So when things are less active, when there's less photosynthesis and deposition from the roots, we find the microbes are breaking down some of those fatty materials. And then things like these organic acids that I talked about that are prevalent throughout the winter, but also early in the growing season. So we see there's a real seasonality to these microbes, and the substrates that they like to use are paired to what's available with the plants. And the really important thing to note here is that even over the winter months in a perennial system, or if you had a cover crop and winter cover crop, we see a lot of microbial activity that can contribute to nutrient availability and carbon storage. Here, if we just look at the exudate metabolites and lipids that are coming from the plants, we can see the metabolites are high in nitrogen here, while the lipids tend to be higher in carbon. And so this is fueling the microbes in different ways and providing all of the necessary nutrients that help to cycle in the soil and then feed back into the plants. We did some isotope probing to look at these metabolites and lipids that were derived from the microbes themselves then, once they're feeding on plant inputs. And we find that many of these pools persist even after a year. So this shows how microbial metabolites and lipids can persist. Those phospholipids that we were showing in the bare soil, this can help to build organic matter. And that anything that's associated with minerals really tends to last for long periods of time, and they're very abundant in the soil. So these mineral components can be really great for building organic matter and supporting activity of microbes for beneficial functions. So together, I hope I've shown you that microbiology is really important for soil health. We see that root inputs can promote microbial growth and stimulate microbial contributions to nutrient cycling and soil organic matter formation by interfacing and consuming with those metabolites. that the microbial growth, activity, and death contribute to organic matter, even during these winter months when we see that microbes are still generating enzymes, they're still living off of some of these metabolites that are less bioavailable and important for carbon storage purposes. We find that microbial residues, so that dead microbial matter, can also build organic matter by contributing those lipids, proteins, and mineral-associated organic matter that we saw persisting in incubations even after a year long. And so finally, perennial plants or winter cover crops are going to be really important for supporting microbial biomass and these beneficial microbial functions. All of this work was done in a big team at PNNL and in collaboration with Washington State University at the IREC field site and funded by the Department of Energy. And so thank you very much.
Thank you all for those excellent presentations, and welcome everyone to the panel discussion portion of the soil microbiology session for SoilCon 2026. My name is Maren Freisen, and I am an associate professor in the departments of plant pathology and crop and soil sciences at Washington State University in Pullman. My research focuses on mutualistic interactions between plants and their microbiomes and how interactions between organisms can be harnessed to promote both plant and soil health. And today I'm really excited to introduce our discussion on soil microbiology. As you just heard in their presentations, our three panelists are experts in the role of soil microbial communities in agricultural soil health. In today's session, we will dive deeper into each panelist's research and extension work by discussing new frontiers in measuring and interpreting soil biological health and practical takeaways for growers seeking to manage their on-farm soil microbiomes for both economic as well as ecological outcomes. So with that, I will introduce today's panelists. Our first panelist is Dr. Jean-Michel. He is a professor in the Department of Plant and Agroecosystem Sciences and in the Department of Bacteriology at the University of Wisconsin-Madison. Through his research, Jean-Michel seeks to improve the benefits of microbes to crops, including the benefits of biological nitrogen fixation to non-leguminous plants. Our second panelist, Dr. Mallory Choudoir, is an assistant professor and extension specialist at North Carolina State University. Her research and extension work focuses on understanding the ecological and evolutionary processes driving plant-soil microbe interactions for informing land management decisions in North Carolina agriculture. And finally, our third panelist, Dr. Kirsten Hofmockel, is a senior scientist in the Biological Sciences Division of the Pacific Northwest National Laboratory, PNNL. Her research focuses on how plant microbe interactions shape soil biogeochemistry, helping to identify key molecular processes that influence ecosystem responses to environmental change. So it's my extreme pleasure to ask all of you to share with our group what is the one most exciting or surprising development in the past roughly 10 years for our understanding of the connections between microbes and soil health. Jean-Michel, would you like to start us off?
Sure. So for me, I would say that probably what was the most novel and surprising was the importance of microbial necromass as a major source of stable carbon and organic matter in the soil. Of course, fungal necromass seems to play a very important role, but also bacterial necromass and the role of, EPS, exopolysaccharides, and other substances produced by fungi, such as mycorrhizal fungi, in soil carbon sequestration.
For our listeners who are maybe less familiar with the term, could you define necromass for everyone?
Sure. That would be the material left by dying organisms. So when fungi die, they leave in particular their cell content, but also in particular their cell wall, which is going to remain for quite a longer time in the soil.
Wonderful. Kirsten, do you want to maybe speak to that or add your own?
Yeah, it's funny. That was absolutely one of the top when I was like, oh, just one. And I was sort of debating between two. So I'm glad you brought up that one because that's a topic that's really dear to my heart. And I think it has really changed how we think about soil organic matter formation, right? Because microbes are so tiny and it's hard to imagine how important they can be for soil organic matter formation. And yet we know that both dead plant material and dead microbial material can build that organic matter. And so understanding how microbial biomass blooms and dies can be really important for building organic matter. And the one most exciting or surprising development that I was thinking of in addition to that, so now I get to do two since I went second, is multi-omic technology. So this is more of a capability, but I would say a decade ago, we were just starting to understand how genomes, like the DNA of microbes, really encode for these functions, all these wonderful functions that they perform, like Jean-Michel's research is on nitrogen fixation, and that's encoded in the genomes of organisms. And so the technologies for us to understand not only the genomes, but then like all of the metabolites, all of the molecules that they're exchanging with each other, and how plants can recruit for organisms that help them, and how, there's a lot of interactions, chemical interactions that are happening between microbes and plants that we can leverage in order to increase soil health. And so I think those capabilities have really changed how we think about the soil microbiome. And it's not just this black box of like, we know they're there, we know they're good, but now we can start to really identify specific organisms and pathways that we want to promote in order to maintain healthy soils.
Great. Mallory, what about you? What's your absolute
Yeah.
favorite?
Okay, my answer encompasses both of your answers. I love necromass. I like to talk about necromass as in we want microbes to live, die, and then stay dead, right? Like stay in the soil as necromass. So my answer is, and I'm going to say this, and it's going to seem like, yeah, of course. But in the last decade, we've really been developing these methods in order to quantify and like demonstrate this like concept I'm going to talk about. So the idea that microbes are modulating their environment, and the environment is also like dynamically changing soil microbiome communities, right? And that seems like, yeah, of course, right? Like microbes are changing their habitat and the habitats are changing microbes. But that's really complex to qualify and even quantify. And it really has been in the last few, years and decades that we're able to show that indeed microbes are adapting and responding to their environment, and then are in turn, through their interactions and their processes, changing the soil environment, whether that's improving soil health or interacting with different plant species. So sort of that like reciprocal connection between microbes and their abiotic habitat.
Yeah. Okay, so maybe we could shift to thinking about what these functions are that we're wanting to promote in our soils. So how might each of you define soil health from the perspective of the microbiome? Mallory, do you want to give us
Ooh, ugh. I've been using the sort of soil Anna Karenina hypothesis, whatever it's called, how there's all sorts of unhealthy soils, but there's no one definition for a healthy soil. So, again, like think about this a lot from an applied perspective of agro-ecosystems, and surely we can identify processes that contribute to healthy ag soils, right? Like improving organic matter content, soil organic matter, aggregation, water infiltration, right? Like microbes can contribute to all of those functions. So that would be, right, like a healthy soil, a soil that can produce crops, that has these like metrics that we know contribute to healthy ecosystems. But it certainly is context dependent, and depends on the location and like the system and what we're asking from the ecosystem to define what is healthy or not.
Yeah. Jean-Michel, what do you think?
So yeah, I agree about the stability aspect and the fact that it's extremely context dependent. So for me, when I think about health, I think about one, a stable state. a state where you have resistance and to change and resilience. So I think about something stable. And the second, I think about a state where that delivers the benefits that you want. And so that's why I think it's so difficult to define health or a healthy microbiome or a healthy soil, because it depends on who you talk to. And I was thinking the same soil, let's say a corn grower versus somebody who would like to do a prairie restoration project, they will expect something different from the same soil. And so for them, health means different functions. So I think it's, to summarize for me, it's a stable state, resistance and resilience. And two, we need to think about the benefits that we expect from that soil. And it's dependent on who we are talking to.
Mm-hmm. How about you, Kirsten?
Yeah, I guess I would offer a different perspective in that I think there are some unifying principles of soil health that probably apply to most soils. And even in the example you gave, Jean-Michel, both of them would aim for plant productivity, whether it's like a diverse prairie restoration or like corn, it's like plant productivity, and that's from a healthy microbiome. And so I would argue that the healthy microbiome, soil microbiome, has a lot of diversity, right? Because that gives it resilience, but it also allows it to be adaptive as conditions are changing. And it's really similar to plants, right? Like you don't want to have if you have a monoculture, it's going to be more susceptible to pathogens. Whereas if you've got diversified cropping systems, you're more resilient and adaptive to climate changes, or to if you have, changes in weather or other stresses that might come into effect. And so I think some of those aspects of diversity, functional diversity, biological diversity, are really important generally for a healthy microbiome.
Yeah. So given all of this complexity, what are some of the ways that you guys think that farmers could be actually managing their microbiomes on farm?
I mean, in terms of management, like I'll say the obvious thing, which is like roots in the ground. Like you have to have roots in the ground all the time.
Mm-hmm.
No bare soil, right? I think winter cropping systems are really important. That's what feeds the microbiome, and it produces the biomass and produces the necromass, and it produces soil organic matter then. So we've got to have like photosynthesis providing nutrition to the microbes. So that's sort of an easy one, is roots in the ground year round.
I totally agree. For me, I was translating roots in the ground by carbon flow, but I think it's a lot about that. Another aspect is also, I think, disturbance, trying to limit disturbance. Growers know that no-till systems are beneficial. And so limiting, in particular for fungi, limiting disturbance, I think is really important. And nutrients can disrupt these communities extensively. And I think limiting nutrients is also part of what we should try to do.
Yeah, we here in the East Coast, we have a lot of really low carbon soils, like very sandy, less than 1% carbon. So in thinking about soil health, right, it really does need to be context dependent. So, here it's, maybe no-till is not really feasible, right? But minimizing disturbance, or I'm not a tillage expert, right? But there's like a lot of brilliant strategies for planting and harvesting and sort of cover crop removal that are minimizing disturbance relevant to your system. Feeding microbes with both roots, right? Roots in the ground, and then also organic amendments is a really good strategy to build carbon. So giving microbes as much food as possible so they can eat, die, and then stay dead. Right. And thinking about, right, any practice where you see soil being lost, if it's like flying up into the air or being like run off in the ditch in the road, like those are the practices that we want to minimize.
Yeah. So for farmers who are interested in learning more about their microbiomes, are there any kinds of measurements that you guys recommend? Should everybody be, Kirsten, you want farmers sending you multi-omics samples
No.
from their farms?
I mean, I think that's for research and development, right? So we can come up with solutions. You know, if there was going to be a rapid test, it might be for microbial biomass, like the active biomass, just to know like how healthy is your soil. That I think would be a relative index though, because to your point, Mallory, we're in aridosols here, and so they're dryland soils. And so again, they're going to be really different than like the mollisols in the Midwest, right? But understanding how your biomass is changing or your live microbiome.
Mhm.
Yeah, I mean, I would love to have a microbiome related indicator for some process that we want to optimize. Um, I don't think we're there from a practical producer farmer standpoint. There certainly are third-party testing companies that provide this type of information. Generally speaking, I don't think there's a lot of actionable outcomes from that data beyond what you should normally do, right? Like diversify inputs and crop rotation and all of the practices that we just talked about. But it's too simplified to say like, yes, there are microbial indicators of ABC, and you should go out and test those.
Yeah.
I think what makes it a bit challenging for growers to implement is that it's a long-term process. It's as you said, Mallory, it's not something which is going to increase yield the following year. And I think many growers are interested in the long term also of their system, but they need to be ready for it's a multi-year process. And I think evaluating carbon, I mean overall, if there is only one in the soil is already a good indicator of how microbes are probably gonna thrive in that soil. And there are many techniques to evaluate different forms of carbon.
Mhm, mhm. Yeah. Does anyone have any final thoughts that they want captured?
I think it's really valuable to have conversations between farmers and folks that are more in the R&D space so that we can come together and try to translate both directions and find, you know, this challenge of are there, you know, rapid tests that farmers could do for the microbiome? And we're like, we're not there yet, but continuing to stretch to think when we are doing our research, are there things that could translate rapidly or be optimized for like a field assay? I think that's a really important goal. And so I value these opportunities to sort of bring us all together, all the different stakeholders. So thanks for the opportunity.
I'll follow up a little bit on Kirsten's. I think it really is an exciting time with like new and developing technologies. So I'm grateful to be working in extension where I have fantastic partners to translate this applied work into not just evaluate, but actually um, like devise the questions that need or that should be answered. So I think we're in a really, yeah, great spot to be, communicating with each other and like developing research across producers, growers, researchers at different types of institutions, and then technology and industry partners.
Okay, last chance. Okay, I feel like maybe I'll wrap it up. Does anyone else have any? Okay, cool. All right. Thank you all for participating in this discussion and sharing more about your work and your knowledge and expertise about this really, really important topic. Yeah, we all, I think, appreciate the outsized role that microbial communities play in addressing soil health concerns, both across regions and agricultural systems. So with that, we'll end today's panel discussion, and I look forward to hearing more questions from the webinar audience now.
Thanks to our speakers for the great presentations and discussion, and thanks to all of our participants for engaging in the Q&A and in the chat. We have a few minutes for questions and answers here, so we're going to get started with that. I'm going to start with one that came in that Mallory answered by typing the answer, but I want to have her give voice to her answer as well. The question was, Mallory, can you say which bacterial community composition is better than others, or are they just different?
Right. So at a snapshot, we cannot look at the members of a microbial community and say this is good or bad in the context of a diverse soil microbiome that we typically see. For this study, we need to do more. In order for this to become this like predictive power, we need to go beyond in this study, in this field, which those microbes will be unique to that field. And there we can say, oh, this particular microbe in this plot, this community was associated with increased SOC, right? Like higher yield in that plot. But that's meaningless when we go on to the literal next field right beside, because there's going to be different microbes there. So it's not that I don't think we can build this knowledge of what sort of microbiome features are correlated with beneficial outcomes. It's not going to be at the level of the specific composition, which is fundamentally different between fields.
Okay, thanks, Mallory. The next question I'm going to direct to Jean-Michel. The question is, how do levels of synthetic fertilizer impact the relationship between the plant and the soil biology?
There are several publications and studies that have shown that synthetic nitrogen fertilizers, first, they suppress the activity of nitrogen-fixing bacteria in the soil, but also in the long term, they reduce the quality of nitrogen-fixing bacteria. And so they impact massively in the short term and the long term the benefits we can get from these bacteria.
Thank you. I will direct the next question to Kirsten. As soil health is an emergent property with processes that result from the integration of physical, biological, and chemical characteristics and processes, how do you integrate your biological expertise into the much more complex transdisciplinary environment of soil?
Mm-hmm. Yeah, this is sort of the grand challenge, right? And even that like complexity, as Mallory was talking about, it varies across the soils. Like if she's in like South Carolina, or I'm here in Washington State, and in the Midwest, we're going to see lots of different kinds of physical, chemical, and biological complexity. And so ideally, we would work across lots of fields. Sometimes that's not feasible because of like funding, basically, or just logistics of it. And so sometimes we'll like compare analyses across groups of scientists to try to understand what's happening across fields and what's driving those responses. Within individual lab groups, though, what we often do is we partner. So we try to work in the field, so we're grounded by what's actually happening. And then we design experiments in the laboratory where we take samples from the field and we manipulate them in the lab in order to have more controlled conditions. And so if we, have a hunch about what's really causing these responses, we can test them directly in the lab where we reduce some of those factors and get down to a simplified system where we can demonstrate the mechanism. And then often we return back to the field. So even in like that nitrogen fixation example that Jean-Michel was just talking about, you could take some of those microbes in the lab and add synthetic fertilizers, and you could remove some of the other variables and just see how they respond and start to get like a dose response curve or something, and then go back to the field and design a field experiment to demonstrate that that's consistent across many plots or many field trials. And so I think that coupled lab to field approach is really important. I think one of the tricky parts of it is sometimes you might drop a factor that's really important. So when we simplify those experiments, we have to be really thoughtful about how we do that so we don't lose one of the control points that are coming from the field.
Thanks, Kirsten. The next question I'm going to start by directing to Mallory, and then Jean-Michel and Kirsten can chime in if they'd like. When starting soil health restoration, humics have flooded the market in Saskatchewan with live bugs, etc. And that's kind of a two-parter, I guess. This part is about humics, and then the next part is about whether having a better balanced base saturation and plant available calcium would elevate the microbiome or improve the microbiome. So we'll start with you, Mallory.
You hit me on not my area of expertise in soil chemistry, so I'm gonna basically pass it off right away. I will acknowledge, yes, the immense market of bugs, jugs of bugs, and right, like humics. But I guess my one comment is the habitat you provide will recruit the microbes that like to be there. And whether that relates to plant productivity, the microbes are happy to do that as long as their other features of their environment are to their liking.
Okay, thank you. We just have a couple of minutes left, but Kirsten, do you have anything you would like to add to that?
I mean, I guess I would just add what Mallory said earlier in the panel conversation too, is that then those microbes also can change their habitat, right? So it'll recruit the microbes that can survive there, and then there might be functions where they alter it. But I think that answer is really hard because the response to the humics depends on like the actual chemistry of that addition and what the chemistry of the local site is as well. So, tricky question.
Okay, anything to add, Jean-Michel?
No.
Maybe they covered it. Okay, great. Let's see. Here's one. I'm going to kind of open it up to any of our panelists who would like to respond. Shelby said, I remember at last year's SoilCon, Dr. Sundstrom mentioned that the microbiome of an ecosystem responds best to the organic inputs it evolved with. What do you think about this in the context of your work or this conversation? Jean-Michel, any quick comments?
It's more an observation that many of the bacteria that we try to grow in the lab and use as a product usually fail to compete with the microbes that are already in the soil, which is in part, I mean, due to that, I guess, the kind of carbon sources that are going to have when we grow them to produce an inoculant are going to be very different from what they're going to encounter in the field. And I don't have a clear, maybe it's not the perfect answer, but I think it's a major issue we have when in the use of biological products in agriculture is the lack of competitivity of the microbes we try to introduce with the native microbiome.
Thank you for that. Unfortunately, I'm going to need to cut it off because we are at time for Q&A, but thanks to all of you for your questions. Our speakers will be able to respond to additional questions in the Zoom chat for as long as they're sticking around for a little while. And we're going to go ahead and move on to our next session.